That's not smart.
I'm a college student, so.
I'm not in place for it. Alright, fair.
Okay, couple of things. How do you feel Monday went? Bad? So
What
some
did it
bad,
drive?
some terribles. I talked to a couple of people who said it wouldn't, not so bad. I haven't even looked at them yet. I was getting ready for lab yesterday, or Monday, and then had lab yesterday. Probably start looking at your exams today, and I might or might not have them back to you on Friday. We'll just see how the grading goes. Luckily, there aren't too many of them, so I should be able to turn them around relatively quickly. One of the things about exams is the better you do on them, the faster they are to grade. So for my own sake, try and do well on exams because it just makes my job easier. I mean, I'm serious. It's so easy to grade just a bunch of correct answers. It's the easiest thing in the world to do. What gave you the most problems on the exam on Monday?
I thought the pictures of the mitosis, meiosis
Yes.
That was hard.
weren't very clear.
Oh, okay. What was unclear about them, do you think?
Just like the sketches weren't like didn't look how they would look in like the textbook or.
PowerPoint,
Okay.
yeah, they did look different than on the PowerPoint.
Yeah.
It's like basically exact same in terms of, I don't know, it was a little bit, I was screwed.
So you were confused by the fact that the chromosomes looked different
Yes.
Okay.
On the PowerPoint
than they did in the thing.
Yes.
Yeah, I actually did that on purpose because I actually thought the ones in the PowerPoint were less clear because they're all wiggly, and the ones that I did on the exam. So that's actually good to know because I'll know not to do that next time. So the ones that are in the PowerPoint.
That's just what we were studying.
Yeah, so mitosis and meiosis, where is it? Oh, here we are. So these were some of them, and then these were others of them.
Yeah.
Because they were wiggly and they're also not the right colors, then I realized the ones on the figure were, on the test were in black and white. What I basically did was I had to draw, let's see, I had to draw these guys. And so these are the guys that were on the test, because I think they're actually clearer in a more schematic way of looking at things. Let me pull up the test real quick. What else did you find confusing or difficult about the test?
The first page was a little, for my own personal experiences, I didn't know exactly what to do, but.
You were supposed to critically think about things and then come up with a nice
Correct.
critical analysis of it.
Correct.
And the reason that I gave you that stuff on the Friday before was because to ask you to do that kind of thinking on the fly in an exam is unfair. So I wanted you to have some time to spend with those graphs so that you would not come to it cold. And then I gave you a bunch of kind of questions that you could use to kind of look at those data on Friday, knowing what I was going to ask you on Monday. But there are lots of things that you can do with those kinds of data. So it was basically kind of the same kind of exercise we did the first day of class. And those kinds of things take time to think about, which is why I gave you the weekend to think about it.
Was it strategic that you purposely like sent that to us on the weekend before the test, or was that just like something that like?
Oh, I sent it to you on Friday on purpose because I wanted you to see it. And what I don't want to do on questions like that is have you walk in and then you're faced with this crush of data on the day of the exam, and you're like, what the hell do I do with this?
Yeah, that's kind of what I did.
So.
Was there one right answer for that, or was like?
I think there's one answer that the data point to.
Which was?
Basically, the EPA has been around since 1971. There have been times when there was a lot of regulatory activity by the EPA, and there were times when there was not much. And if you look at periods of heavy regulation and the periods of low regulation, in the Obama administration, there was a lot of regulation, and coal jobs were tanking. In the Reagan-Bush years, Bush 1 years, there was considerably less regulation, and coal jobs were tanking. In the second Bush years, there was not very much regulation, and coal jobs were going up. Coal jobs go up and down for a lot of reasons. EPA regulations are probably not one of them.
Is the price of coal one of them?
Certainly the price of coal is one of them. So the price that you can get for your commodity influences whether or not a company is going to go into that sector. And so those of you in business, business majors, you know, if the price of something is going up, that's a signal that there's a market there for that, and so people enter that market. Did we have touchscreen cell phones before the iPhone came out? The answer is no. The most advanced thing we had was a, what do they call those things that the government used?
Blackberry?
Blackberry. We're Blackberries with the little keyboard that you typed on. Apple came out with a touchscreen iPhone, and they had the market for about a year and a half cornered, and so they could charge whatever they wanted for an iPhone because there was nothing else out there like that. And then what immediately started happening?
Android.
Every company on the planet started going for touchscreen iPhones, and now I challenge you to find an iPhone that isn't a touchscreen iPhone, a cell phone that isn't a touchscreen cell phone. They're all touchscreen cell phones now. So you see a good market there, and everybody starts jumping into the market. So coal employment expanded during that time when prices were going up because it's like, oh, let's get more coal. The thing that started killing coal jobs that is not on any of those graphs is I didn't plot for you, although I could have, was the rise of natural gas in electrical generation. So right now, about a third of our total electricity output is coal. 50 years ago, almost everything we put out electric, in terms of electric power generation, was coal. Some of it was hydroelectric, but the bulk of it was coal. And it's because when fracking became a thing, fracking is a way of getting at oil in shale deposits, but one of the byproducts of fracking is it generates and releases a lot of natural gas from those same deposits. And natural gas has become more and more abundant, so the price of natural gas has plummeted. Natural gas actually burns cleaner than coal, so electrical companies have been closing down coal plants and opening up. Every time they close a coal plant, they do that because they're bringing on a natural gas plant, because natural gas is cheaper to buy and it's also cleaner burning. And so coal has been declining in terms of demand because natural gas has become so cheap with the rise of fracking. And the other thing that has happened, the other thing that I wanted you to get out of that figure, what happened with coal production and coal employment during the mid-80s to now?
They were inverse of each other?
They were inverse of each other. You're getting more and more productivity out of, in terms of total coal production, while you're also cutting coal mining jobs. What happened there?
Technology.
What kind of technology? Do you know?
I'm not exactly sure, but I'm assuming they figured out a better way to be more efficient and productive.
They figured out a way to get more coal with fewer coal miners. And what that happened was in the late 70s and early 80s, they shifted from having coal mining done mainly in underground mines, which are very labor intensive and also dangerous. And they moved to mountaintop removal, where you just dynamite the top of a mountain off, expose the seam, and then scoop it out with these gigantic cranes. And that takes relatively few people. You need some people, you need a crew of people who are good at demolitions, and then you need a couple of crane operators and a couple of dump truck drivers. And so it was a much more efficient way of getting a lot of coal out of the ground, required many fewer coal miners. And so a big part of the decline in coal mining employment was actually this change in coal mining technology, taking coal miners out of holes in the ground and putting them on the surface. Once you do that, you need many fewer of them. Yeah, you had a question.
So what was the presidential, it had each president on the line, what was that for?
Just so you would have a reference, so
Was there any significance that like a president was affiliated with this part of the?
no, the main thing is some presidential administrations were more activists in terms of utilizing the EPA than other administrations were. So like Reagan was not very environmentally active. Bush 1 was not very environmentally active. Obama was very environmentally active. Carter was. Strangely enough, Nixon was a Republican, and he signed the bill that formed the EPA. So it's not so much party affiliation, although that was on there as well, as much as seeing that there were presidents who saw this as more of a priority than other presidents were. But then when you look at coal and the number of people employed in coal, those things aren't actually correlated with one another at all. You know, you had, let's go look at that.
That was my answer right there.
Was that your answer?
That was my answer. I assumed that there was some correlation to any of that since it was on the graph.
Well, so things are on the graph sometimes because they
Just
mean
to throw it
something,
out.
and then they're also there because they don't mean anything. Where is that?
They're just on there.
They're just on there.
There's no way to tell like what they were doing though, so.
So is that usual that you'll throw us an extra thing just to be critical and have to figure it out on our own sense, like the weekend before a test? Is that something I should be aware of?
No, so one of the things that a CTI class should be teaching you is how to critically think about things. We're going to be talking about genetically modified organisms in agriculture for test 2 material. You're going to read a National Academy of Science report on GMOs. So the National Academy of Science is this group of scientists. They're independent of the government. They're independent of the party. They're basically just the smartest people in the room if the room were the United States. And occasionally they get charged with studying certain issues, and they wanted to look at genetically modified organisms because genetically modified organisms are becoming more and more prevalent in agriculture, and people have concerns about them. So they wanted to take an academic approach to looking at the issue of what are the effects of genetically modified organisms. So this morning on the Moodle site, I put the National Academy of Science report. All of us should read chapter 3. Chapter 3 is kind of an introduction in history of GMOs. And then I'm going to break you into groups, and each group is going to be assigned chapter 4, 5, and 6. Chapter 4 looks at agronomic and basically generally agricultural issues. Basically looking at GMOs from the perspective of a farmer and getting yield out of your crops, et cetera, et cetera. Also looks at what the effects might be on pest communities and things like that. Chapter 5 is about human health. And chapter 6 is about socioeconomic effects. And so, economic effects. And then on October 4th, we're going to come together, and each group is going to make a 15-minute presentation about their chapter. We will have all read chapter 3, so we'll have some basis of common knowledge, and then each group is going to inform the rest of the class on kind of what their chapter said. Because after that time, you're going to sit down and write a position paper, and the format of the position paper is imagine that you are at Thanksgiving dinner, and while you're eating the bountiful abundance of Thanksgiving dinner, somebody mentions something that they heard on the radio about GMOs, and you have to take a position on whether GMOs in our food supply is a good idea or not. Because it's quite frankly your Aunt Bertha who's making these statements, and you as a student have read a report provided by a bunch of scientists who don't really have any investment in the game, so to speak, who have tried to evaluate GMOs in our food supply objectively and scientifically. And so you know something about the things that Aunt Bertha is saying. And so your paper is essentially your Thanksgiving letter to Aunt Bertha, either correcting her of her misconceptions or providing support for her misconceptions, depending on what it is that Aunt Bertha is saying. So it's a position paper, but it's a position paper that should be written in a way that anybody not a scientist could understand what it is that you're talking about. And it will be I'll ask you to touch on these three major components. What do these things mean from the perspective of a farmer? What do these things mean from the perspective of human health? And what are the socioeconomic ramifications of these things? So that's gonna be a written assignment that we do probably at the beginning of November or something like that. So we'll do the discussion of it at the beginning of October. Since you will not all have read all three chapters before you write a paper about it, you might want to go back and read the chapters that your group didn't read, but at least at that point you'll have kind of a group's summary to help you get an introduction to that chapter is kind of how it will work. So that's going to require critical thinking on our parts to get through that report. So there's going to be a critical thinking part of probably every exam. The critical thinking that we did on the first day of class was about how to use data to do this, which is why I then gave you another data set. So here's the figure, and you can see coal production going way up while coal jobs are crashing. Strangely enough, during the Obama administration, a lot of regulation and coal jobs increased before they then declined. Once again, this decline is largely due to changes in technology in the oil sector, fracking that was releasing a lot of natural gas, which was driving down the price of natural gas. Back here, this is Nixon Ford. Oh, Nixon Ford, a lot of regulations. Why are there a lot of regulations in the Nixon Ford era?
I don't know anything about politics.
The EPA was just formed. Like you make this cabinet level post, cabinet level post, and as a result of that, this post is new. The first thing that they do is they do a lot of things. So you have a lot of regulation early on, and coal jobs are skyrocketing during that time period. Carter was very environmentally sensitive, so there's a lot of regulation during the Carter administration. Once again, during Carter's term, coal jobs are going way up. Reagan comes on, very little regulation during Reagan, Bush 1, and also Clinton was not particularly environmentally active. And during that time of relatively low regulation, you see that coal jobs are crashing, essentially. Going down, but once again, this decline in coal jobs is happening at the same time that you're getting an increase in coal production. That's indicating that something's going on with technology of coal extraction.
But if you look at the graph below it, during that time of the drop, the price of coal is also
is declining.
Mm-hmm.
Why does the price of coal decline?
Because they're
What drives prices of commodities?
they have more technology to get more coal, therefore they have more supply.
You have way more supply, that's then driving the price of coal down.
Mhm.
When coal gets cheaper, what do power companies want to do? Buy more coal, build more coal-fired plants rather than natural gas plants. And so many of the coal plants that were built back here in the 80s, the lifespan of an electrical coal power generating plant is 30 to 40 years. A lot of those things are becoming obsolete. They just get old, they wear out because you're generating tons of heat, metal becomes stressed, et cetera, et cetera. So the lifespan of any electrical producing facility is about 30 years. As those old coal-fired plants are becoming obsolete and being decommissioned because they're no longer functional, the power companies are replacing them with natural gas plants because now natural gas is much less expensive than coal is. So a lot of coal being used, a lot of coal-fired plants being built, as those things become obsolete, they're being replaced. So this low price during a period of time when employment is going down is largely being driven by super production because of a shift in coal extraction technology. So yeah, Republicans and Democrats both did a lot of regulation. Coal jobs soared. Republicans and Democrats did very little regulation. Coal jobs dropped. So the question on the exam was, to what degree does EPA regulatory activity influence coal jobs? The answer is, well, with a lot of regulation, coal jobs went up. With less regulation, they went down. And then with more regulation, they went up again. But then they were also going up during the second Bush term. Very little regulation by Bush, but coal jobs are going up again. It just doesn't have anything to do with regulation. Yeah.
So do we get a point if we said that there's no correlation?
Yeah, I mean, that's exactly what it says.
Okay.
Like,
The extra, like, critical thought process.
well, you need to explain to me why. I mean, you can't just say there's no correlation. You needed to explain why there wasn't a correlation. Why you think that's the case? Yeah. Okay.
What if you give how many points if we kind of read it the complete opposite of what is actually right, I guess?
Well, you would have to give me a good quantitative reason
Okay.
for believing you.
That's I did, okay. Cool.
Yeah, I mean, if you can convince me that the data are showing something different,
Yes.
then yeah, I'll give you credit for that. I think it's going to be kind of hard to do that because the data are the data. This is the
I think I just completely misunderstood what the truth.
okay.
Yeah.
This is the
Yeah.
this is the irritating thing about being a scientist. I'm liberal. I'm on the liberal side of things. I think the EPA is great. When I was a kid, we were polluted as hell. So in my lifetime, the Cuyahoga River has burned. Like a surface of a river was on fire. You guys know about the Cuyahoga River? Oh my gosh, you guys don't know about the Cuyahoga River?
That's crazy.
Let's all Google the Cuyahoga River. We're not getting to genetics this way. I always have trouble spelling Cuyahoga. Cuyahoga River Fire, Ohio. Okay, the Cuyahoga River has burned a number of times. This is the 1952 fire. That is a picture of a river flowing through Cleveland. And the surface of the river is so polluted, a spark set it on fire, and it just basically burned for days.
How long ago was this?
This was 1952, but it happened again in 1969. So it happened in 52, they didn't clean it up then, it happened in 1969. Guess what happened in 1971? The EPA was formed. The EPA was formed as a direct result of people seeing the Cuyahoga River burn on television. And so when I was a kid, I was born in 1966. When I was a kid, we had pretty much thrashed the environment in the United States. When I was a kid going through public school and we would have a section in social studies or something on environmental science, we talked about acid rain. When I started teaching, when I was in graduate school in the early 90s, there were, if not big sections of the section on nutrient cycling about acid rain, there were entire chapters in environmental science textbooks on acid rain. Did anybody talk to you guys about acid rain when you were in public school? They did? Go look at an environmental science textbook nowadays. You don't even see people mention acid rain. You know why?
It doesn't have
The regulation.
Acid rain is not a problem anymore. Acid rain was caused by burning things like coal in manufacturing plants, mainly in what's become the Rust Belt, the area around the Great Lakes. When you burn coal, it releases sulfur dioxide. You take sulfur dioxide, combine it with water in the atmosphere, and it turns into sulfuric acid. That stuff blew into the Atlantic states, mid-Atlantic and North Atlantic states. It got so bad that it was eroding the limestone buildings in New York City. If you go to old cemeteries in the upper portion of the Atlantic seaboard, you can't read tombstones anymore because they've basically been weathered, and that weathering of the tombstones was hastened by the presence of acid rain. The Environmental Protection Agency was formed by Congress and signed into law by Nixon, and they were tasked with cleaning up air and water in the United States. And they required manufacturing plants to put what we call scrubbers inside of smokestacks. So all of these Smokestacks from factories throughout the country had to be retrofitted. A scrubber is kind of like your catalytic converter on your car. Your catalytic converter basically removes lead from the emissions from your car. And so there were just these screens that had a catalyst on them that removed sulfur dioxide. And over the course of like a decade, acidity of rainwater turned itself around, and we went from acid rainwater to neutral pH rainwater. And so, you know, I remember as a kid, you know, driving into Houston, there was just like this layer of smog about 2/3 of the way up the tallest skyscrapers in Houston. And Houston is still by no means a clean city, but you don't see those smoggy days nearly as much as you used to. My first job when I was an undergraduate, my first research-oriented job, I worked on a project funded by the Forest Service looking at the effect of acid rain and ozone on trees. Like, acid rain was a thing. And you don't hear anybody talking about acid rain as an issue in the United States anymore because it's just largely not an issue in the United States. So I'm a big fan of the EPA. And I'm a big fan of presidents who are environmentally active. And I would love to say, you know, all Democratic presidents are bad and all Republican presidents are, or all Democratic presidents are good and all Republican presidents are bad. But you know what? On the environmental front, I can't really say that. I think Nixon was a douche. But he signed into law the Environmental Protection Agency, and his administration passed a ton of environmental regulations. Part of that was due to the fact that the agency was new and Congress was paying attention to this because people didn't want to see the Cuyahoga River catch on fire again. But the fact of the matter is he could have vetoed those bills. But you know what? He didn't. I would love to say Clinton was a great guy. He was actually kind of a douche too, for a different set of douchey reasons. But in terms of environmental regulation, he really didn't do much. So in answer to your question, Caden, my problem as a scientist is I have to look at data and I have to let the data tell me what to think. And so I could paint with this broad brush and paint all people of one type bad and all people of another type good. But if I then go and look at the data about those people, I have to do what the data tell me. And the data tell me that environmental regulation doesn't really have anything to do with coal jobs. Coal jobs are influenced by something else out there in the world. And as a business major, you should know that what drives everything job-wise. Supply and demand, in this case of a workforce, and changing technology also influences workforces, and those are going to be much more powerful forces in an economic system than something like environmental regulation. And the environmental regulations that get passed when different parties are in power is just kind of willy-nilly. And I can't figure out why Clinton was so lax on environmental regulation. I know why Obama was so active, because he had an agenda. But Clinton, I think Clinton's agenda was kind of just getting along and being liked. And you don't want to piss people off when you want to be liked, and so you don't do things like pass regulations. Bush was an oil man. He came from an oil history. I mean, he was an oil executive before he became president. No surprise that he had a particular look on the EPA. But, you know, I don't understand Nixon and Ford. They were just on this front. People who I think were probably responding to the kind of tenor of American upsetness at the state the environment had gotten to at that point. And here's the thing. So in 2017, and I put this in the question, there were 13 bills introduced into the House and Senate designed to abolish the EPA, either to strip it of any regulatory power, or there were a couple of bills that were actually entitled the bill to abolish the EPA. 13 bills in the House and Senate to get rid of the EPA. And I think one of the things that has happened in the United States is you guys have grown up in a world, in the United States at least, that's pretty clean. Part of that is due to the EPA's activity, part of that is due to technological advances, but a lot of those technological advances are driven by regulation, as it turns out. And so you don't think the EPA is important because you actually are living in a world that is made better by the presence of the EPA. You don't know that because you didn't grow up when I grew up, when basically things were not so good back in the day. So one of the nice things about having things good is you get complacent about things, and then you say, well, why is this EPA in existence? The environment's clean and the EPA is killing jobs. So then you can ask, well, why is the environment clean and does the EPA actually kill jobs? And the only way to ask that question sensibly is to look at data. You can listen to Democratic presidential candidates and Republican presidential candidates talking about this, but you know what they never do?
Reference the data.
They never show you a graph of actual data. You, as critical thinkers, if you and when you engage people in the political realm, you should always be saying, show me the data that shows me that what you're saying is actually true. If they're unwilling or unable to do that, you know what you should not do? You should not believe a word that comes out of their mouth. Critical thinkers are skeptical, and critical thinkers are governed by things like data. So Caden, I don't know what you wrote on your paper for this question. I'm going to look at your answer, I'm going to compare it to the data, and if your answer conforms with data, I'm going to grade it correct. And if it doesn't conform with data, I'm going to say, what were you thinking? Data should drive us. And occasionally you have to let go your preconceived notion. What was the preconceived notion that I had to let go on the first day of class?
That we would actually know what we're doing here.
No. No, that is not the preconceived notion that I let go of. What were we talking about the first day of class? We were talking about the price of soybeans, actually. Thank you for playing. Talking about the price of soybeans and what thing that's going on right now in the national political.
Terrorists.
No, not terrorists.
Terrorists.
Oh, sorry. I thought you said terrorists. Sorry, tariffs. Yeah. So the tariffs on China and soybeans, and farmers are complaining a lot about Trump and tariffs and the price of soybeans. The price of soybeans is slumping because China is going elsewhere to buy soybeans. They're not buying as many American soybeans, so we have this glut of American soybeans. And if you talk to farmers, the sky is falling. And I would love nothing more than to have all of these farmers who voted for Trump be pissed at Trump next year because soybean prices are down and vote Trump out of office. But when you look at the data, guess what? The tariffs are having a tiny, tiny, tiny effect on the price of soybeans. So while I think a tariff war, a trade war with China is a really, really bad idea, I don't think it's really negatively affecting farmers in any great degree compared to, once again, these broader issues that affect the price of farm commodities that really are beyond the control of Trump or anyone else. So I would love nothing more than to see Trump drummed out of office and to have Midwestern farmers do it. But if Midwestern farmers are doing it because the price of soybeans are down, they're not engaging their brains at all. Because the price of soybeans hasn't really changed that much since the tariffs have been in effect. Not nearly as much fluctuation in price due to tariffs as due to other things like drought and years of plenty, which then goes back to basic economic issues like supply and demand. What's the supply of soybeans relative to the demand? Droughts have a much greater influence on those prices than any sort of, in this case, a government regulation applying a tax to imports on Chinese goods that then causes China to place an import tax on American goods the first thing that China put a tariff on was soybeans. Why? Because they knew that farmers voted for Trump and they thought it was gonna hurt farmers. And while it is having a small effect on farmers, it's not a catastrophic effect. They've experienced we showed that they experienced way bigger fluctuations in price over the last 40 years or whatever that graph showed than what the tariffs are doing. So while I would love to jump on the Trump's an evil dude because he's killing farmer income, I can't really fault Trump for the trade war on that particular front. Yeah.
I think I saw something a couple of days ago that China was backing off the tariffs from the soybeans.
Yes. So China's backing. So the whole tariff thing is a moving target. Things are constantly changing because Trump is now saying, "Oh, we're gonna, back off of our tariffs on China." So China's saying that they're gonna back off their tariffs. It's just a weird form of gamesmanship. And I think what markets ultimately like is stability. And the reason markets are responding poorly lately is there's a lot of instability in knowing I told you that farmers are actually ordering seed now for next year. It would be nice for them to have some stability to know, well, are these tariffs still gonna be in place? In which case, I'm not gonna plant soybeans next year, I'm gonna plant corn. Because I know where I can sell my corn, and I can sell that corn domestically to ethanol producers and things like this. So that's that. So I had to look at the data and say, okay, farmers are saying one thing, and I want to buy into what farmers are saying. Is what farmers are saying actually true? Farmers seem to be a little misinformed, probably because they're relying on their memory, and in their memory, prices for soybeans have been much higher, but those high prices for soybeans were much higher because of the drought when soybean supply plummeted, and as a result, prices went way up. And we plotted where droughts were on that plot. And so the problem with our memories is our memories are faulty, and we tend to remember the recent things that we've seen rather than the way back things that we've seen. And prices have been much lower for soybeans in the lifetimes of most farmers than what they currently are. But we had these highs during those droughts of the early to mid 2000 teens that drove prices of both corn and soybean way up. And so we have this incorrect view that soybean prices have always been really high, and they have been really high in the past, but they've also been really, really low in the past as well. So data destroys misconceptions. So yeah, going back to the whole mitosis meiosis thing, the thing that I wanted you to pay attention to is the number of chromosomes. There's 4 here. Because there's 4 here, that means that you're early on, you're in prophase. They're not lined up along a midline anywhere. So you're in prophase, you're either in prophase 1 or you're in prophase 2 because you still have 4 chromosomes. Sorry, you're either in prophase 1 of meiosis or you're in prophase of mitosis because you still have 4 chromosomes. Why is this meiosis, not mitosis? Because those homologous pairs of chromosomes are matched up with one another and they're going through the process of synapsis, they're crossing over. So you can say, okay, well this is now prophase, but it's prophase 1 of meiosis. In this case, we have 2 cells. Each cell has how many chromosomes?
4. It's 2.
Chromosome 1, chromosome 2, chromosome 1, chromosome 2. Each one has 2 chromosomes. Now, it's a chromosome that's been duplicated such that it's a chromosome that exists as 2 sister chromatids, but you only have 2 chromosomes, so we've undergone a reduction division, so this is either the end of meiosis 1 or the beginning of meiosis 2, because you see that you now have half the chromosomes that you began with. Yeah.
So the spindle fibers start to move out, and try to attach to stuff during prophase, is that correct?
Yes, they kind of grow out from the centrioles. I didn't talk to you about centrioles because they're not that important, but these spindle fibers grow out, and then they make contact with the chromosomes, and they're actually responsible for ordering the chromosomes here along the middle. So the important thing is to count chromosomes, and if you're at 4 chromosomes, you're either in mitosis or you're in the first phase of meiosis. And then as you go through a reduction division during the first phase of meiosis, you begin to see the differences between meiosis. But you can see the differences between meiosis and mitosis all the way back in prophase. And I don't have a prophase of mitosis. Why is this metaphase of mitosis and not metaphase of meiosis? Metaphase 1 of meiosis.
They're not crossed.
Yeah, your homologous pairs aren't hooked up with one another. They're gonna, when they go to partition chromosomes to new cells, you're gonna split the centromere and divide the two sister chromatids and put these two sister chromatids in different things. Here we have something going on. In this big cell, how many chromosomes are there? These were joined together, these were joined together, so that's 2. When this ends up getting all the way over here, you're gonna have 2 chromosomes here, 2 chromosomes here, 2 chromosomes here, 2 chromosomes here. So are you doing mitosis or meiosis here? You're meiosis because you've undergone already a reduction division. You've reduced the number of chromosomes from 4 to 2, which means that you're in meiosis 2. And since these are being pulled apart by these spindle fibers, you know that you're in anaphase 2. So it's all about the chromosomes. If I had not given you what the diploid number of chromosomes is, it would have been harder to figure all of this out. So I just give that to you to help you figure that out. So it's all about chromosome count. And sorry if these chromosomes were confusing to you. They're actually simpler to me in my mind because they're more schematic and less natural looking. So other questions about the exam? Next test is going to be just like this. You're going to have some problems to work out, some genetics problems, because we're going to be talking about genetics. You're going to have to know some processes, so the way in which we genetically modify organisms involves some molecular biology. I'll walk you through that to the best of my ability. I try and give you only the details that you need to know in order to understand the process without getting too much into the weeds on that. But it requires you to know what the structure of DNA is like and stuff like that. So some of the stuff that is test 1 material will also be on test 2 material because we continue to talk about genetics. Yeah?
What was the answer to number 6?
Carbon dioxide and water, 2 simple molecules come together to produce glucose, water, oxygen, yeah. I do a lot of things like that. So you see that what did I do on the test? I did the same thing that I did in class. Can you make a table out of something that allows you to compare and contrast 2 things that are supposed to be compared and contrasted? What did I do? Why did I do that in class? Well, I did that in class because I knew I was going to do that on the test. It's an easy way to organize knowledge, and so I want to make a test that makes you give me back knowledge in the most organized sort of framework, because by asking you for that, hopefully it will also encourage you to go through this organization of knowledge before you get to the test, which is also why I introduced ways of organizing this knowledge in class so you can use those things. In this way, I'm kind of teaching to the test. Yeah, you have a question?
We should expect every test to be just like fill in the blank, no word maker like that.
Well, it's not all fill in the blank. There are some sentences and stuff like that, but the degree to which that I can organize the test, that I keep the task of taking the test clear for you, then you'll do better on the test, hopefully. It makes the test easier to grade, in a sense. One of the problems with essay tests is students get really rambly. So on the first page, what did I tell you at the beginning of the exam?
You'll let it be alone.
Hmm?
You just gotta answer the questions that you have on there.
There's a lot of space there. Don't feel like you have to fill the whole space. Answer the explicit questions that I ask. As soon as you've done that, move on. What I saw when people turned that in is they filled the space. And the more you fill the space with information that is tangential to the questions that I've asked, the harder it becomes for me to grade that. Because you told me something that's wrong, but it doesn't really address the question directly. How do I then grade that? Or you tell me something that is right, but it doesn't address the question directly. Do I give you credit because you knew something that actually isn't relevant to the question? it becomes harder for me to grade those types of questions objectively. So even on questions where I'm asking you for sentences or maybe a brief paragraph, I want you to always be going back to the question. One of the other things that I do, and I do this in all of my classes, is oh, I didn't do it on this one. I should have done it on this one. I did on question 8. So I have a big question here, but it's actually a big question that I've broken up into a series of smaller questions, because I know that if I just ask you the big question, what's my big question in question 8? Explain the differences between rubisco and carboxylase. How does this difference affect drought resistance? How does that relate to stomatal opening? And how do these things relate to water loss? What am I asking you in this question?
You didn't open it before.
What's the difference between C3 and C4 photosynthesis? But if I asked you that big question, what is the difference between C3 and C4 photosynthesis, I would have gotten a lot of different answers. And so I have a question here that's 4, 8, 12 points. If I had said, describe for me the difference between C3 and C4 photosynthesis as it relates to plant water balance, God only knows what I would have gotten. And then there would have been this 12-point question that I get widely different answers about, and then I don't know how to grade it, and quite frankly, you don't know how to answer it in reality. It's just too big and too broad. And so what I do is I say, okay, if I was going to ask that big question, what are the subcomponents that I want to know if you know about that get to this big thing about water balance? Oh, well, you need to know the difference between rubisco and carboxylase, because that's how carbon gets fixed. That's ultimately what affects the plant's need to get new carbon dioxide into the leaf. That affects stomatal opening, and that ultimately is what affects water balance. And so rather than ask you this big, broad question without any elaboration, I ask you a series of smaller questions that then tells me whether or not you know the answer to this big question. Yeah.
So back to the very first question, how will you end up grading the first question, since it is 10 points? That's like a huge
Oh,
amount.
yeah.
How will you end up grading that?
I probably wrote that question that way because I wanted you to be a little more freely thinking. I'm going to grade that question on the basis of what's the strength of your argument, and is that argument based in data. So Caden might convince me that his answer is right as I go through and read the answer. And so that's what the purpose of that question is, which is why I didn't bust it up into a bunch of little subcomponents, because I want you to feel free to freely critical think about that. There's no critical thinking here. You either know how C3 and C4 photosynthesis affect water balance in plants, or you don't. Does that make sense? Yeah, so on the first question, I can be swayed towards believing what you're saying, but it will depend upon the strength of your argument and how well supported that argument is by the data that I've given you.
But there's still like a correct answer versus like a not correct answer.
A correct answer.
Because
A correct answer is an answer that conforms to the data, ultimately. And if you can convince me that your answer conforms to the data as you're interpreting the data, then yeah. So on those graphs, if Caden drew lines going up to really demarcate things that he was paying attention to, so that he's looking at really narrow slices of the graph or something like that, and that convinces me that, okay, because you're parsing the data really thinly, you come to a different conclusion than perhaps I would have come to a conclusion, because I'm looking at it more broadly, then I'm perfectly open to those kinds of arguments. But ultimately, the purpose of those kinds of questions is to ask, how do you think when you're confronted with a bunch of data?
So if I were to portion it up in a specific way to prove a point that even if I look at it broadly is still incorrect, I could prove this one point.
One of the nice things about
points.
one of the things that's nice about a 10-point question is there's always room in there for partial credit. So I have no idea what you people wrote on that question. I'm gonna have to read those things and figure out for myself, okay, what's the strength of your argument? Am I being convinced by your treatment of the data? And if I'm convinced by your treatment of the data, then you're good. If I'm unconvinced by your treatment of the data, I'm gonna probably take the points off. And then at that point, if you feel that you need to come and convince me about the strength of your argument, you're free to do that. So, yeah. One of the things about critical thinking is I think a room full of critical thinkers would be able to look at a set of data and come to the same conclusion ultimately, regardless of what your particular frame of mind and biases were coming into it. One of the things about critical thinking is that you recognize what your biases are. I've been really free with you telling you what my biases are. My biases are I'm liberal. I think government does lots of things like pave our streets and provide police protection and put out fires at our houses. People think taxes are bad. Taxes are just a way that government earns money, raises money to provide services to the populace. I don't like the fact that so much of my tax dollars go to fund the largest military industrial complex on the face of the planet. But you know what? I pay my taxes every year. Partly because they'll throw me in jail if I don't, but partly because it's my role as a good citizen to do that. I don't have children. I pay property taxes on my house and my car. Those property taxes largely go to support the public education system in Liberty, which is quite good from what I hear. You know what? I wouldn't know. You know why? I don't have any kids enrolled in it. I could sit at my house down in the historic district and say, why am I paying property taxes? I don't even have kids. And then I would be a douche. Because somewhere way back when I was a public school student, guess what? Somebody paid their property taxes so I could go to a good public school and get a good education that got me into college, it got me to graduate school, and got me where I'm right now teaching this class at Williamsville College. Property taxes are good because they pay for the police, they pay for the schools. You know what? I would be happy if they raised my property taxes if we could give teachers a raise so that they don't have to take summer jobs. I would gladly have you raise my taxes. Because I think public school teachers are the one thing that separates us from chaos. A well-educated populace will keep us from just devolving into chaos. And we get public school teachers dirt cheap in this country. And we should pay them more. And I'm passionately an advocate of public school teachers. I think we pay them too little, and I think we give them way too much guff. You want to raise my property taxes? Go ahead. I'm good with that. I'm really clear with you where I'm coming from. Now, if you see in the data that my bias is getting in the way of what the data show, your job is to call me out on that and say, okay, those data don't mean that. That I have to step back and look at that data and I'm saying, okay, my bias got in the way of my objective treatment of the data. And I'm good with that. I did that with farmers and soybeans. I'll do that with Republican presidents who were relatively environmentally active. I'm all about giving credit where credit's due. That's what I have to do if I'm going to call myself a critical thinker. We've talked now way more about the first exam than we should have today. All right, I'll try and get these graded and back to you. Hopefully by Friday, we'll see how grading goes. We're talking about genetics. So, we were talking about monohybrid crosses. Monohybrid crosses are pretty straightforward. I'm going to expand your genetics problem set some to give you a little more practice, especially as we get into more complicated crosses. But this first cross is pretty easy. We went through it last week on the board. Basically, you can divide this up into steps, and my steps in working genetics problems is always start with parental generation. Sometimes you'll be given the phenotype, what the organisms look like. sometimes you'll be given the genotype. If I'm given a phenotype, I always try and figure out, okay, well, what would the phenotype be? What are the possible genotypes from that phenotype? So if we have round seeds and wrinkled seeds, I know from before that round seeds are dominant and wrinkled seeds are recessive. So what I know, if I'm just given these phenotypes, is wrinkled has to be recessive, and round can either be homozygous dominant or it can be heterozygous. One way of shorthanding that is just to put a blank here for what I don't know. But I know that it has to have at least one dominant allele because it has round seeds. We cross those, and if this is a homozygous dominant, this individual can only make one type of gamete. This individual can only make one type of gamete. So all of the F1 offspring will be heterozygous. They'll all be round. So after you've dealt with the parental generation, the next thing you need to do is you need to make gametes. That's what those are. I always circle my gametes to remind myself that that's a gamete, not a genotype. And then the act of fertilization is just combining those gametes into a new genotype of the next generation. If, on the other hand, we observe that half of the offspring are round and half of the offspring are wrinkled, then that tells me that this individual could not have been a homozygous dominant. It had to be a heterozygote. It'll produce two types of gametes, a gamete containing the dominant allele, a gamete containing the recessive allele. We put this into a reduced little Punnett square, and we get a heterozygote for one offspring and a homozygous recessive for another offspring. Half of the population is round, half of the population is wrinkled. So whether I give you a genotype or whether I give you a phenotype, what comes out of crossing those two individuals will give you some insight into what the genotype is if I haven't given you the genotype. In this particular example, we have true breeding parents. The true breeding parents can only produce one type of gamete each, and so as a result, we get a heterozygote. All of those dudes are round seeded. Yeah.
Real quick, I'm sorry. Gametes are the like big R, little r, or
Yep. So this is why we went through meiosis. So remember in meiosis, an individual has a cell, let's say that only has one chromosome pair. So it has only two chromosomes. Its diploid number is 2. 2n number is 2. This is chromosome 1 of this set of chromosomes. If it is a heterozygote, when it goes through the S phase of the cell cycle, it's going to duplicate the DNA there. So you'll have two sister chromatids. This one has an R, this one has an R. It will have two sister chromatids after duplication in S phase, and this one will be little r, because this one gets duplicated and this one gets duplicated. Now when you go through meiosis, the first phase of meiosis occurs where these two things synapse. But since we're only dealing with one chromosome type, if you get crossing over here, let's just say that we do get crossing over here, this will now be a little r chromosome at the tip, this will be a big R chromosome at the tip. But then these guys get drawn over to one cell, these guys get drawn over to the other cell, so that at the end of meiosis 1, in telophase 1, you have a chromosome here that is big R, little r, big R, little r. They're separated, they're connected by a centromere. I'm having trouble drawing on the side. Big R, little r, and you have the same thing happening here, connected by a centromere, big R, little r. This is telophase 1. As you go through metaphase and anaphase 1, this chromatid gets drawn to this side, this chromatid gets drawn to this side, this chromatid gets drawn to this side, this chromatid gets drawn to this side. So you end up with 4 cells at the end. Each cell has 1 chromosome. 2 of those are big R, 2 of those are little r. There's only 2 gamete types. There's a big R gamete type, there's a little r gamete type. So if you have this genotype, you can only produce 2 types of gametes. The big R gamete types and the little r gamete types. There's 2 of each, but it doesn't matter. There's essentially half of the population of gametes is 1, the other half of the population of gametes is 2. So why draw 4 of them when you can just draw 2 of them? Keeps the Punnett square simpler. So that's what this individual produces. This individual only has 2 little r gametes, 2 little r alleles, and so they can only produce the little r gamete type. Because they start out like this. When that gets duplicated, all of this is little r, so when that all gets split apart, everything's little r. Does that make sense? Yeah.
So you said true breeding
Mm-hmm.
is,
When I say true breeding, it means that when they self-fertilize, they'll produce the same genotype and phenotype as they are. And the only time that you get that is when you are homozygous.
okay.
So these guys produce the 1 gamete types that they can. They produce 1 offspring type. They're round, they're heterozygous. If we then cross their offspring with one another, in the case of peas, we just leave them alone and let them self-fertilize. What happens is now each of these individuals can produce 2 gamete types. This guy can produce the big R, little r. This guy can produce the big R and little r. And when we put those into a Punnett square, we get one homozygous dominant, 1 homozygous recessive, and 2 heterozygotes. All 3 of these individuals here will be round, and the wrinkled will be 1. But we have 3 genotypes. We have homozygous dominant, we have heterozygote, we have homozygous recessive. There's 1 of these, there's 2 of those, or there's 1 of those. This is the phenotypic ratio, this is the genotypic ratio. And this is how it works with 1 trait. What we're gonna do on Friday is we're gonna add a 2nd trait. So we'll look at round and wrinkled seeds, and we'll look at something else in combination. Oh, we'll look at seed color. So the next thing we'll do is we'll add, are you round or wrinkled, and are you yellow or green? And then we'll see how that changes how all of this works. It doesn't really change anything, it's just that we now have to keep track of 2 traits rather than 1 trait. And then after that, we'll add a 3rd trait and see what that does. And that's Friday's problem. Questions?
What does that lead to? I know that we're always going back to the omnivore's dilemma and how that progresses our further knowledge.
So the section of chapter 1 at the end where it talks about pork sex. This is color of seeds in wheat. It's shortened by 3 genes, as it turns out. In order for you to understand how hybrid corn gets made and why farmers have to buy hybrid corn, we get to this figure that is in your textbook.
Is the food journal due Friday too?
Yes, 3 journals are due Friday.
Blogs are due Friday.
We're working up to this. This is how we get hybrid corn. These are all standard Mendelian crosses in order to understand what Monsanto does and Cargill does to get a hybrid corn that it then sells to farmers. You have to know this, and in order to know why farmers don't just save seed from one generation and plant it the next generation, once again, you have to know this. The reason that they don't save seed is because those seeds are actually now crosses between hybrids, and all of the desirable traits that they paid for in the hybrid seed are now diluted as a result of crossing hybrids with one another. So in answer to your question, all of this is leading up to this thing, which is the second to the last slide in this long presentation that we've been working on. So it is headed somewhere.
Yeah.
You just have to be really patient to get there. I'm laying the groundwork for you understanding what this is. I'm also laying the groundwork for you understanding the discussion about GMOs as well. That all builds into this.
Perfect. Thank you.
So don't lose sight. I have my eye on the ball. Yes, ma'am.
What kind of cells produce gametes?
The cells in your ovaries and the cells in my testes, and in plants, it's the cells in the anthers, the male part of the flower. Remember?
So in male parts, it's ongoing, but in
Okay, so in plants, you have a stamen and there's an anther at the end, and they all get made in the anther, and it's not ongoing. It gets done once for the most part, but they make gajillions of them. And in the ovary, it happens once, and there are usually fewer of them. Male and female, I think I already told you, male and female, defined by biologists, is determined by gamete size. The egg is always the bigger gamete, and it's always referred to as the female gamete. The small gamete is, in this case, when we're talking about humans, it's sperm, and that small one is always the male gamete, so that's how we kind of assign sex in something like a plant. It doesn't really have sex the way we think of it. So male flower, female flower part, because these guys produce small gametes, these guys produce giant gametes. In humans, it's a weird thing. Dudes are constantly producing new sperm every day. They produce them. If they don't get used, they die off, they get recycled. They get broken down, all the proteins and everything there, and then get recycled. But we're producing new sperm every day. Females are interesting.
because of one of all the.
Yeah, so you start off with your stem cells that are ultimately going to go and make gametes. They go through prophase 1. They get to metaphase 1 when you are a fetus, and then it stops. And then every month when you go to ovulate, your body picks one of these cells that is at metaphase 1 and says, you're chosen. It's totally random. And then at that point, it starts up again, goes through anaphase 1, and goes through all of meiosis 2, and produces 4 eggs that you typically ovulate one of them, because the other eggs get used for other things as you're developing follicles. And so every time you go to ovulate, you take one of your cells that has been arrested at metaphase 1, back when you were a fetus, and then it kicks off and continues through the rest of metaphase. So you don't produce them every day, you produce them every 28 days approximately, but they've all started the process back before you were even born, which is freaky deaky actually.
So gametes themselves don't just go through mitosis instead of having the whole meiosis thing.
Gametes don't go through mitosis, they go through meiosis.
Right, and that's because of the genetic variation. Because if you had just a cell with 23 chromosomes and you wanted more of those, why would it not just go through mitosis? Why would the gamete itself not? I'm assuming it's genetic variation.
So essentially, mitosis 2 is essentially just a mitotic division. So meiosis 1 is this special kind of division because the homologous chromosomes match up, and that results in a reduction division. But then meiosis 2 is just mitotic division of those reduced genome cells. What happens, so this is easier to draw on a male. So males have tubes all through their testes, and at the base of this tube is a ring of cells, and at the base of that tube, these cells are all diploid, and they all have a complete nucleus. And what happens happens at the beginning of spermatogenesis when a male goes to make sperm is the first thing that happens is this thing mitotically divides. And what it does is it produces two new cells that are now genetically identical to that cell, and then it's this cell, these two cells that start undergoing meiosis. So these are the it's been so long since I've taught anatomy.
Like a template or something?
Yeah, there's a special name for them. Oh, spermatogonia. This is why you should not become a scientist. So these guys are cells that are kind of the primordial cells that then produce the cells that go on to become sperm. Because if this guy underwent meiosis, he wouldn't be diploid anymore.
Yeah.
And so he has to produce some diploid cells that will then go through meiosis. Otherwise, you'll run out of cells, especially as many sperm as males produce. And so males can keep producing sperm because they always are undergoing mitosis to produce these cells, and these cells are then the ones that go through meiosis and then produce sperm. And it's actually weird, there are these other cells that provide nourishment for the sperm cells, and these cells actually kind of crawl through the cytoplasm of this cell, and this cell, which is called a nurse cell, gives nourishment to these guys, because as these guys go through meiosis, they get smaller and smaller and smaller, so that at the end, a sperm is basically just a packet of DNA, a packet of chromosomes, a little sheath here that has mitochondria in it, which are the things that generate energy. Mitochondria, the little organelles that generate energy, and a flagellum. There's hardly any cytoplasm in these things, so they lose a lot of their function. They're basically just a rocket. They're a rocket powered by a flagellum and a mitochondria, and the warhead is a packet of chromosomes. And at the cap of this thing, there are enzymes. And when the female creates an egg, the egg is surrounded by layers and layers and layers of cells. Many layers of cells, sometimes 6, 7, 10 layers of cells. And it takes millions of sperm crashing into this layer of cells. When they do that, it ruptures this thing that have digestive enzymes in it. It kills some of these cells, and they slowly sacrifice themselves on the follicle cells that surround the ovum, and they slowly burrow in by releasing their digestive enzymes that kill these cells that are protecting the egg, until finally one comes into contact with the membrane of the egg, and that's when they get to fertilize it. And so males produce tons of sperm because they have to get past the defenses of the egg by basically releasing these enzymes, and all they are really is a little missile full of chromosomes. And they don't really have any function other than to fertilize an egg. So your mitochondria that you have came from your mother. My mitochondria that I have came from my mother. So all of those organelles that we have actually are found in the egg, because the male sperm doesn't contribute anything to the new zygote that's formed after fertilization except the DNA of the male. Classic male. Males are just male. Yeah.
Okay, well, thank you.
Yeah, so it's really kind of fascinating. I haven't seen a good biological explanation as to why females have arrested mitosis as fetuses. Probably because I haven't looked, but I don't know that anybody has a good answer. Somebody might have a good answer, but that's one of the interesting things. And that's also one of the problems with having births later on in life, is these cells that are designated for being turned into ova, they've been hanging around since you were a fetus in this arrested stage. They're not produced anew from a new bout of mitosis that then produces cells that go through meiosis at the moment that you need them.
Right.
So if you're 45,
It's a little dusty.
if you're 45, the egg that you produce at 45 is an egg that's been sitting around waiting for 45 years. So we have, when people have babies, when women have babies later on in life, there are oftentimes more complications, and part of it is that these aren't fresh, newly divided cells with newly replicated DNA. It's really old sure, no problem.
See you on.
Luckily, I know some things about things other than food.